The following learning module, Cell Transport, will provide a brief general overview over cell transport content material to prepare for the biology end of course tests. Please complete the Cell Transport Learning Module review sheet as you proceed through the learning module. First of all, let's talk about the cell membrane. The cell membrane is a barrier that separates the cell from its external environment. It forms a boundary between the cell and its outside environment. The structure of the cell membrane includes a double layer of phospholipids. Please bring your attention to the enlarged diagram of a phospholipid in the bottom right-hand corner. Phospholipids include a hydrophilic head, meaning water-loving, and a hydrophobic tail, meaning water-fearing. This provides the rationale for the arrangement of phospholipids in the cell membrane. The tails point towards the inside of the cell, while the hydrophilic heads point towards the outside of the cell. Other components of the cell membrane include cholesterol molecules, which provide strength of the phospholipid bilayer, carbohydrate chains, which serve as identification tags, and protein channels, which assist molecules with transport. We'll talk a little bit more about protein channels as we proceed throughout the presentation. In addition, it also controls what may enter or exit a cell, hence why the cell membrane is considered semi-permeable. Some things can enter and exit the cell without energy or assistance, while other items will require energy and or assistance to move throughout the membrane. The cell membrane basically selects what may enter and exit the cells. On the following slides, I will discuss the various ways molecules may cross the membrane. Molecules move across the membrane because of concentration differences. Here are some important terms to remember when referring to cell transport mechanisms. Concentration is the number of molecules of a substance in a given volume. Concentration gradient, which you will hear and reference a lot throughout the presentation, is the difference in the concentration of a substance from one location to another. The mentioned terms will be important to consider when determining the movement of particles across the membrane. Materials move across the membrane because of concentration differences. Fortunately, some molecules may enter and exit the cell without requiring the cell to do work. This type of transport is called passive transport. The movement of molecules across the membrane from areas of high concentration to areas of low concentration. In terms of passive transport, and thinking of the energy mechanism, think of passive transport as riding a bicycle downhill. No energy is required as you ride a bicycle downhill. In terms of concentration gradients, think of perfume being sprayed in the front of a room. As time elapses, the perfume sits and spread out across the room, leaving the area where there's a high concentration of perfume molecules and spreading out to areas of lower concentrations. Three types of passive transport that will be discussed in this review module include diffusion, osmosis, and facilitated diffusion. At this point, pause the video and record your own example of passive transport on your review sheet. Now let's refer to the Venn diagram graphic organizer located on your review handout as I go through the three types of passive transport. First, let's talk about diffusion. Diffusion is the movement of molecules from areas of higher concentration to areas of lower concentration. The key difference is the material being transported. Molecules are transported during diffusion. The next type of passive transport I will discuss is osmosis. Osmosis is the movement of water from areas of higher concentration to areas of lower concentration. Here, the material being transported is water molecules. Important to note that. The last type of passive transport I will discuss is facilitated diffusion. Facilitated diffusion is the movement of molecules across the membrane through the use of transport proteins. The molecule is transported with the assistance of a transport protein. Refer back to when we talked about protein channels when we were discussing the makeup of the cell membrane. All three types of passive transport have the following in common. They all involve the movement across the membrane from an area of high concentration to an area of lower concentration. This movement will continue until dynamic equilibrium occurs. On the previous slides, I discussed how some materials are able to travel across the membrane without the use of energy and down their concentration gradients. In contrast, some materials require the cell to use energy to transport materials that cannot diffuse across the membrane. This type of transport is called active transport. Active transport occurs when a cell uses energy to move molecules against a concentration gradient. Molecules are moved from a lower concentration to a higher concentration. In terms of active transporting energy, think of active transport as riding a bike uphill. Energy is required as you travel up a hill, and more than likely you will break a sweat traveling up an extremely steep hill. It does require energy input from the cell, and three types of active transport that will be discussed in this review module will include endocytosis, exocytosis, and the sodium-potassium pump. Now let's refer to the second Venn diagram graphic organizer located on your review handout as I go through the three types of active transport. Endocytosis is when a cell membrane engulfs a substance and brings it inside the cell. You could think of it as cell eating. Exocytosis is the opposite of endocytosis. It involves the removal of substances out of the cell by diffusion of a vesicle with the membrane. Both endocytosis and exocytosis transport material across the membrane in transport vesicles. Lastly, the sodium-potassium pump. This involves the movement of sodium and potassium ions across a membrane through the use of a pump. It requires energy in the form of ATP as well as a carrier protein. The mentioned three types of active transport have the following in common. They all require energy and move particles from an area of low concentration to an area of high concentration. Energy is required and the movement goes from low to high. Now let's refer to the understanding check on your handout. Now we will check the understanding of the cell transport mechanism. The blue molecules have a higher concentration gradient on the outside of the cell than the inside of the cell. The arrow indicates that the molecules are moving down their concentration gradient from an area of high concentration to an area of low concentration. Therefore, this model represents passive transport. More specifically, it represents diffusion, and no energy is required here. Now pause the video for a moment and complete the following two examples. Then resume the video when you are ready to check over your answers. The second model we'll discuss contains sodium ions. The concentration of the sodium ions is higher on the outside of the cell than on the inside of the cell. The arrow indicates that the sodium ions are moving upwards against their concentration gradient. The ions are moving from low to high. So this is an example of a sodium-potassium pump, and energy is required. The last model with the clustered red molecules also represents a higher concentration of molecules outside the cell than inside the cell. The arrow indicates that the molecules are moving down their concentration gradient from an area of high concentration to an area of low concentration with the aid of a protein channel. No energy is required, and since there is a protein channel there, we know that this must be facilitated diffusion. You have now reached the reinforcement activity for this learning module. Please access the PowerPoint, double-click on the cell transport word puzzle and the cell transport word find. This will open the PDF file and allow printing of the following documents. Thanks for watching this learning module. The following sources were used in creation of this presentation.